lymphocyte activating killer cells, tumor reactive cytotoxic T cell injected with interleukin 2. What's the LAC cell? It's tumor reactive cytotoxic T cell injected with interleukin 2. So it is cytotoxic T cells that attack the tumor cell injected to the patients with cancer in association with interleukin 2. And this is the LAC cell. This is a patient with CA, and we collect its blood, about 30 cc, and then separation of the lymphocyte from his blood, and start to culture it by tissue culture with anti-CD3 antibody, and then there is a proliferation and amplification of these T cells, and then collection of these lymphocytes, and then reinfusion to the patient in association with interleukin 2. And here, when we culture the lymphocyte, culture it with interleukin 2 to stimulate proliferation of these cells. The another type of the tumor immune therapy is using of TIL cell, which is tumor infiltrating lymphocyte, injected with interleukin 2, and it's had a wide range of the anti-tumor activity, and it's more toxic. It's need long period of time, 30 to 45 days. And here the patients with CA lung, and we extract and remove the tumor mass by surgery, and we took from this tumor mass the lymphocyte that infiltrate the tumor tissue, and then culture it with interleukin 2, and then reinfuse to the patients, which is more cytotoxic to tumor cell. And this is called TIL cell. TIL, it means that we remove the lymphocytes that we infiltrate the tumor mass and culture it with interleukin 2 to become more active and more proliferative and more toxic, and then re-injected to the patients with tumor. And this cell will be specific and directly attack the tumor and residual cells that stay after surgery or chemotherapy or radiotherapy. There is immune checkpoint. Immune checkpoints are regulators of the immune system. These pathways are crucial for cell tolerance, which prevent the immune system from attacking cells. Inhibitory checkpoints molecules are targets for cancer immune therapy. So these immune checkpoints are present normally in the person, because it's the benefit of this is to reduce tolerance, and we can use this method to treat to be a source of immune therapy against the tumor. Due to their potential for use in multiple types of cancer, currently approved checkpoints inhibitor block cytotoxic T, lymphocyte antigen 4, and PD1 and PD ligand 1. PD1, which is a programmed protein 1, is also known as PD1, and CD279 is a protein on the surface of cells that has a role in regulating the immune system response to the cells of the human body by down-regulating the immune system. As you remember that the PD1, which is the second, the inhibitory signal to T lymphocyte activation, and promoting self-tolerance by suppressing T cell inflammatory activity. This prevents autoimmune disease, but can also prevent the immune system from killing the cancer cells. So here the antigen presenting cell, and here the activated T cell, and this is the CD28 receptor that's bind to the 7 family ligands, which is the second signal in the T lymphocyte activation, and this is the inhibitory molecule to the second signal of the T lymphocyte activation, and this is the PD1 with PD ligand 1. So this is the checkpoints, 2 checkpoints. And this is the tumor cell, and this is the T cells, and this is the dendritic cell, and this is the dendritic cells with the T cell, and this is the checkpoint, and here the checkpoint, and this is the first signal in the T lymphocyte activation, and this is the second signal in the T lymphocyte activation, and here the plus mark sign, and here the minus sign, that this is indicate inhibition to T lymphocyte activation, and here minus sign, that means this is the second checkpoint that inhibit T cell activation. So we have 2 checkpoints. This is the second signal that inhibited CTLA4 that inhibit the second signal of the T lymphocyte activation, and this is the second checkpoint that also PD1, PD ligand 1, that inhibit the T lymphocyte activation. So this is normal. While in the tumor, where there is tumor cell presence in the circulation, so this is the first signal in the T lymphocyte activation, and the tumor cell will produce this checkpoint that inhibit the T lymphocyte activation from attacking the tumor cell. So the immunotherapy will act on these 2 checkpoints, this first checkpoints, and this is the second checkpoints. So programmed death ligand PD ligand 1 is a transmembrane protein that has been speculated to play a major role in suppressing the immune system during particular events such as pregnancy, tissue allografts, autoimmune disease, and other disease states such as hepatitis. Normally, the immune system reacts to foreign antigens that are associated with exogenous or endogenous danger signals, which triggers the proliferation of antigen-specific CD8 T cells or CD4 helper cells, and the binding of the PD ligand transmits an inhibitory signal, as explained before on the figures, that reduce the proliferation of the antigen-specific T cells and decrease apoptosis in regulatory T cells. This is another figure to demonstrate. This is the checkpoint PD ligand, and this is the B7 molecule with CD28, which is the second signal in the T lymphocyte activation, and the inhibition of this second signal by the CTLA4. Several human cancer cells expressed high level of PD ligand. Blockade of PD ligand reduce the growth of tumor cells in the presence of the immune cells. At the same time, it was concluded that PD ligand or B7 helps tumor cell evade the anti-tumor immunity. In 2003, B7 was shown to be expressed on myeloid cells as checkpoint proteins and was proposed as potential target in cancer immunotherapy in humans. So checkpoint inhibitor therapy is a form of cancer treatment immunotherapy currently under research, and the therapy targets immune checkpoints, key regulators of the immune system that stimulates or inhibits its action, which tumor can use to protect themselves from attack by the immune system. Checkpoint therapy can block inhibitory checkpoints, restoring immune system function. And the first anti-cancer drug targeting the immune checkpoint was ipilimumab, cytotoxic T lymphocyte antigen 4 blocker approved in the United States in 2011. Other checkpoints inhibitors target the molecules PD ligand is the PD1 is the transmembrane programmed cell death protein, which is also called CD279, which interacts with PD ligand, or CD274. PD ligand on the cell surface binds to PD on immune cell surface, which inhibits immune cell activity. So among PD ligand function is a key regulatory role on T cell activities. It appears that cancer induces upregulation of PD ligand on cell surface that inhibit T cell activation. Antibodies that bind to either PD or PD ligand and therefore block the interaction. And here, this figure shows us this is the T cell, this is tumor cell, and how the antibodies bind to PD ligand, so inhibit of this checkpoint. And here, in this figure, the antigen presenting cell, here the T cells, and this is the first signal, and this is the second signal, will stop by the antibody inhibitors, and this lead to amplified anti-tumor T cell response, inhibit tumor invasion, migration, inhibit tumor angiogenesis. And the last one, gene therapy, tumor cell or antigen presenting cells or T cells are transfected with B7 genes or genes encoding with interleukin 3, interleukin 2, or tumor necrosis factor. And this is the tumor cell or antigen presenting cell or dendritic cells or T cells, and transfected with plasmid have genes that code for interleukin 3, interleukin 2, tumor necrosis factor. And this will bind with the T lymphocyte, and this enhance the proliferation of T cells. Thank you.